We're in workshop 8, in vivo models and proof of concept trials. First of all, I'd like to start by thanking the Cystic Fibrosis Foundation for providing support for this workshop and for the line of investigation that is being presented here and for the opportunities for us to build on one another's observations and insights. I'm Bruce Schultz, one of the co-chairs for the session today. I'm a professor of physiology from the Veterinary College at Kansas State University. And I'll turn to my co-chair and allow her to introduce herself.
Yes, and I'm Katie Boyne. I'm a pediatric pulmonologist at Lurie Children's Hospital and assistant professor at Northwestern University in Chicago.
All right. By way of housekeeping, there is a very good app that seems to be working well that allows everyone to submit questions as they come to mind using the app. We'll try to monitor the questions as they come in and convey them appropriately. By the way, these sticks in the middle of the aisle are not microphones. Please don't talk to them. It doesn't help us. All right. So it's been 30 years since the advent of the first animal model for cystic fibrosis, the cystic fibrosis mouse. Or should I say the cystic fibrosis mice, since there were a number of different models that were all developed at about the same time. And much has been learned from these animal models and the availability to do types of experiments that let us see CFTR in a more natural or more holistic environment. So today we're going to build on observations that have been made using animal models to gain special insights. When we look back, we see those CF mice and we think those were pretty darn simple by today's standards. But yet, when you see them in the context of what they were, when they were, those were groundbreaking. And so what we're looking at today is just yet another generation of groundbreaking work that's being done. So we're going to work through a number of different animal models that are present. And we're going to start off, Well, first of all, there are no disclosures from the podium here. We're going to start off looking at work that's being presented by Finn Young from the University of Iowa, where they're using a ferret model, but it's a ferret model not per se with CFTR being changed, but rather to look at cells that express high levels of CFTR to see where they're at in the body, to learn a little bit about their lineage and their expression. What is exciting for me is that these cells have been seen and talked about for a few years now in the context of the airway and the lung and the pulmonary tree. But I believe we're going to see them expressed in other areas of the body as well. And so with that, we will learn much about how the body works. Moving to the next lecture, we'll hear from Elizabeth Kramer from Cincinnati, not using a CF mouse per se, but again coming back to the mouse model and using it with a system to knock CFTR out of only selected cells and only at selected times. So we can then understand the specific role that CFTR plays, in this case in the airway, and in how the airway responds to the environment that is present at the time. Moving to that, we'll then move to yet another animal model, here a non-human primate model, the macaque monkey. And as we think back on the history of cystic fibrosis research and curing the disease, we think very much about gene therapy and bringing the gene to fruition to be expressed in cells that are damaged by its absence of expression. And so we'll look at a commercially developed vector to deliver genetic material to the airway and look at the success that that vector is now having in this animal model. Moving forward to the fourth presentation, the rat model will be shown. And this is not a rat model of cystic fibrosis in the knockout sense, as was the first rat model, but rather this is a humanized form expressing G551D, the second or third most common variant of CFTR that is observed, one that's very responsive to potentiator treatment, to the ivacaftor treatment. But what is special about this presentation is not to look at how potentiators increase the activity of this CFTR variant, but rather how it can be boosted even more. And so we'll hear about the use of small molecules to boost this activity even more. Then finally, we'll come back to the ferret model, and we'll look at the natural course of the disease, or perhaps the unnatural course of the disease as you think about the effects of potentiators, as you think of how they can work in a G551D ferret, and then what happens to the tissues if you then deprive them of this great therapy that we now see as being the standard of care, and see how this changes the physiology in the airway. So with that, I'll turn it over to my co-chair to invite the speakers to the podium.
Great, thank you. So we'll start with Feng Yuan from the University of Iowa. If you can come on up. Thank you.
All right, it is my great pleasure to share you guys about my story. So my name is Feng Yuan, and I'm from John Engelhardt Laboratory. So today my topic is single-cell RNA sequencing reveals pulmonary ionocyte subtypes in the proximal ferret airway epithelium. There are no relationships to disclose regarding to my presentation. All right, so first let's have an overview of the CFTR expressing cells. As we know, the proximal airway are composed of basal cells, secretory cells, and synaptic cells, and the CFTR expression was thought heterogeneously expressed across all the cell types. However, in 2018, there are two Nature papers came out, and both papers discovered a new cell type called pulmonary ionocyte, and both papers thought pulmonary ionocyte is the major resource of CFTR expression. And for the pulmonary ionocyte, there is a key transcription factor called FOXI1, and this transcription factor can help to help basal cells specify into the ionocytes. The ionocytes compose about 1% of the surface proximal airway epithelium, but express 50% of CFTR mRNA. So we're thinking about what are the functions of pulmonary ionocyte and how can animal models help to address this question. And our laboratory has been successfully generated multiple cystic fibrosis ferret lines. So here we hypothesize that ionocyte ablation caused by FOXI1 gene knockout can make changes in the airway epithelium function. So here, then we're trying to generate a FOXI1 knockout ferrets by using a CRISPR-Cas9 gene editing technology. As I showed here, we generated a few FOXI1 knockout ferret lines. Now, after those lines are generated, then we bred those lines to get a FOXI1 knockout ferrets. As we know, kidney intercalated cells are pretty similar to ionocytes, and kidney is the most abundant expressing the FOXI1 gene tissue. So we evaluate the FOXI1 protein expression in those knockout ferrets. What we can see here is that the FOXI1 protein expression was absent in the knockout kidneys. And then we move to the immunofluorescence staining. We pick a kidney interkidney cells marker ATP61G3. As we can see in the immunofluorescence staining, the interkidney cells was completely absent in the FOXI1 knockout ferrets, which tell us we already have this interkidney cells were ionocyte ablated ferret model. And the other thing we observed is the kidney tissue. As we can see, in a FOXI1 knockout kidney, the kidney is pretty like watery, which indicates the absence of the ionocyte can cause like a fluid reabsorption issue in those kidneys. So after the ionocyte ablated ferret model established, now we started to test whether ionocyte depletion will affect pulmonary airway differentiation and anion transport as well. So here we use total tracheal cells derived ALI, early culture system to test those hypotheses. So first we tested the synthetic cells marker and the mucin marker staining confirmed the well differentiation of the basal cells derived culture from both wild type and FOXL1 knockout group. And also the FOXL1 knockout cultures demonstrated decreased key ionocyte markers like FOXL1, ASL3, BSND. And we also observed the CFTR expression was reduced in FOXL1 knockout cultures as well. However, there's no change in other cell type markers like synthetic cells marker and secretory cells marker. FOXL1 knockout basal cells derived epithelia showed absent ionocyte transcription factor expression and decreased CFTR expression. And FOXL1 gene knockout does not affect synthetic or goblet cells differentiation. Then next, we move to the electrophysiology to test whether ionocyte absence affect the trans-epithelial anion transport. So here we can see the polarized FOXL1 knockout cultures demonstrated decreased coronary current and bicarbonate current as well, as we summarize in the bar graph. And there's a little change in the trans-epithelial resistance in the FOXL1 knockout cultures as compared to wild type. So pulmonary ionocyte influence anion current in the polarized tracheal epithelia from ferrets. So now we are thinking what other airway properties are affected by ionocyte absence. So the decreased anion transport would predict mucociliary clearance impairment. So here we introduce a mucociliary clearance method here by depositing a gallium tracer into the distal end of the trachea and then monitor the tracer moving from the distal to the proximal end, as we show the image in the wild type. We can see in the FOXL1 knockout ferrets trachea, they show the impaired mucociliary clearance, which is very similar to those G551D ferrets when those ferrets are VX770 off. However, when the G551D ferrets with the CFTR modulator VX770 on, we can see the mucociliary is pretty similar to those wild type ferrets, which demonstrating that the mucociliary clearance is dependent on the CFTR function. And we have the dot plots to summary the mucociliary clearance function, as we showed, like the FOXL1 knockout ferrets showed pretty similar mucociliary clearance to the cystic fibrosis ferret. So FOXL1 knockout ferrets have reduced the trachea mucociliary clearance defect. So what new ferret genetic models can help us to inform those ionocyte functions? So here we generated two ferret line. One is the Rosa TG reporter, which can convert from the tomato fluorescence to the EGFP when Cre is expressed in our cell types. And the other one is the FOXL1 Cre-ERT2, which we insert the Cre-ERT2 gene into the 3' UDR of the FOXL1 gene. So the double transgenic ferret of the Rosa TG and FOXL1 Cre allows us to lineage tracing ionocytes. So here we have the image to show the lineage tracing results. So first we observed the kidney intercalated cells, which is kidney ionocyte, are traced as we showed in the image. And we also observed ionocytes in the surface airway epithelium in the assay in there. And also we observed the ionocytes in some mucosal glands. More interestingly, we see the tracing ionocytes in the connecting ducts. Now we move to an RNA scope to validate the tracing efficiency. As we showed, this is a RNA scope image showed the CFTR mRNA, FOXL1 mRNA, EGFP mRNA co-localization in the ionocyte in some mucosal glands. So then we are thinking, how can such an extremely infrequent cell types have such a large impact on anion transport? I think single-cell sequencing would be the best approach to. One is to define the ionocyte genetics. Two is to check the gene alterations when in those ionocyte-ablated cultures. So here we provide different cultures from wild type FOXL1 knockout, which represent the ionocyte-ablated culture, and FOXL1 Cre Rosa TG, which represent the lineage tracing. So we can on the lineage tracing samples, we also did a FACT sorting to enrich those EGFP positive ionocytes for sequencing to enhance the 10x machine capture ionocyte efficiency. So finally, we retained 77,000 cells for the downstream single-cell analysis. So what we found here is from the single-cell clustering. So we found 8 distinct cell clusters. We annotated those cell clusters based on already existing mouse and human epithelial cell type signature. We call it a secretory basal, cyclin basal, and so on. And we can see the ionocyte cluster in the middle. Then next, we are going to establish a single-cell transcriptomic profile of the ferret ionocytes. So first, we observed that the key ionocyte transcription factor ASL3 and the FOXI1 gene is restricted in the pulmonary ionocyte. And we also observed the other ADP6V1 family protons, which is highly expressed in ionocytes. And the CFTR is highly expressed in ionocytes. We also observed very little CFTR expression in the rest cell types. Then next, we move to immunofluorescence staining from the biology and to evaluate our sequencing results, which we picked a key ionocyte marker ADP61G3 by staining. So from the staining, we can clearly see those green dots are the pulmonary ionocytes scattered on the epithelium. However, this is a high-mic ionocyte image, as I showed here. Now we move to the FOXI1 knockout trachea. We didn't observe any ionocytes in the FOXI1 knockout trachea, which is consistent with FOXI1 gene being required for ionocyte specification. So in our initial data set, we got about 0.1% ionocyte as compared to those published data set. So this is kind of undercounting the ionocytes and promote us to sequence more ionocytes. So the next step, we're going to sequence more lineage tracing ionocyte. Here, so we fact sort those labeling ionocyte. So as I showed here is the EGFP positive, and the tomato positive will be the non-ionocyte. So we mix those cell types at various ratios, like 1:1 to 1:20, then go to downstream single-cell RNA sequencing. And what we found here is once the recovery rate of the pulmonary ionocyte is pretty poor, about 3.4% as compared to those tomato positive non-ionocytes, is about 51%. So now we are thinking about why did those EGFP positive ionocytes go during the sequencing process. So we have there are two possibilities. One is the ionocytes are nest and resulting in the release of the AB and RNA. And the other possibility is the lineage tracing method could label other cell types. Now we tested the first possibility about AB and RNA hypothesis. So first we check the secretory cells which were thought dominating the CFTR expression by other groups. So what we can see here is the ionocyte key marker CFTR, ASL3, and FOXI1 gene expression in secretory cells is positively correlated with the proportion of ionocyte we loaded into each 10x channel. And then on the other hand, we have the perfect model. The FOXI1 knockout is a 100% ionocyte ablated epithelium model. Now, what we see here is the proportion of the CFTR positive basal cells, secretory cells, and synaptic cells from those ionocyte ablated cultures showed a significantly reduced as compared to a wild-type culture. And all those results tell us the fragility of ionocytes likely lead to their undercounting and can increase the AB and RNA levels for highly enriched genes, including CFTR. Now, we're going to test the second hypothesis. Could our tracing method enable other cell types? Here we combine the pulse-seq and EGFP field mapping method. So now we started to use tamoxifen to label those cultures at a pretty early stage. This is a single-cell UMAP. As we can see here, the EGFP expression is in all the real cell types, including neuroendocrine cells, tougher cells, and ionocytes, which indicate those cells at some point use FOXI1 gene for specification. And then we have a dot plot to summarize the results. We can see the EGFP kind of label all the real cell types. Now we move to the biology side to validate the results we have from the single cell sequencing. So first we have the two tracing protocol. One is to label trying to label the cells in the early stage. What we can see here is some of the ionocytes were labeled, but we do see other cells are not labeled ionocytes. So this cell is pretty much, the morphology is pretty much like neuroendocrine cells. Now, on the other hand, on the later stage labeling method, we got 100% of the labeling cells are ionocyte, which is ATP61G3 positive. And here we go through a bar graph to summarize the early stage labeling. We got about 40% labeling cells are ionocytes, and then later stage we got 100% ionocytes. So FOXI1 is expressed during the in vitro basal cells specification of tuft, PNEs, and ionocytes. So we're thinking, would there be a common real cell progenitor exist? Then we go back to the already existed human and mouse real cell type data set. We put all the ferret, human, and mouse real cell type data set together. So we can clearly see the distinct tuft, PNEC, and ionocyte. And also we do see a real cell progenitor in the middle is pretty obvious. And then we did a differential gene expression. This is a volcano map to show the real cell progenitor with other cells. We can see the gene expression signature is very unique and different. So the real cell progenitor exists and also show a unique genetic signature. So then we're moving back to a species comparison specifically on pulmonary ionocyte. As we can see, this is a bubble plot to show the gene expression signature. As we can see, the ferret ionocyte compared to the human and mouse, and the ferret ionocyte, our data set generated is more comprehensive on all the ion channel compositions. And then we did a species-to-species comparison. So this is a correlation from human to human, human to ferret versus human to mice. And what we can see here is the ionocyte genetics is pretty, from ferret is pretty similar to human as compared to mice. So human and ferret ionocytes were significantly more similar than mouse ionocytes. So then we move back to the ionocyte clusters. I think different ionocytes has been observed in other species like fish and frog skin epithelium. So here we recluster our ferret pulmonary ionocyte. We can see those ionocytes can partition into 3 distinct clusters. We name it type A, type B, and type C. And we can see the gene expression signature from the different subtypes are pretty distinct and different. For example, type A highly express the SLC12A2, which is an NKCC channel, BSND, and SAN1, which is INAC channel. And the type B highly express some like immune modulators like CD74, and type C are more like progenitor cells. They express a lot of aquaporins. Then we move to RNA-scope to validate all the different subtypes. So here we observed the NKCC channel and FOXI1 CFTR expressed in the same ionocyte, which is type A ionocyte. We also observed like long SLC12A2 expressing cells, which we think is type B or type C. And we also observed cartin-7, EGFP, and CFTR positive cells in those lineage tracing samples, which is type B ionocytes. And more strikingly, we also observed those kind of proliferating ionocytes, which is very different from the original thought that ionocytes are the terminal differentiated cell types. All right, so a summary from my presentation. Real pulmonary ionocytes appear to have the previous unrecognized subtypes, and human and ferret ionocytes are more significant and more similar than mouse ionocytes. FOXI1 is expressed during the in vitro basal cell specification of all the real cell types. And despite of the low abundance of the ionocytes, they contribute significant amount of the anion conductance in the proximal airway. And also pulmonary ionocytes are important in mucociliary clearance in the proximal airways. So this is all my presentation. Acknowledgement, I would like to thank my mentor, John Gohard, who invented all those transgenic and genic transgenic ferret models and cystic fibrosis ferret models as well, and on the whole ENAP group. And also thanks my bioinformatics arm on this project, Dan Kathik from Broad Institute and Adam Haber for leading some of the ionocyte genetics study and the funding resource as well. With that, I will be happy to take any questions.
Great, thank you. And if anyone does have any, they can enter in the mobile app. We can read them, or if you want to just project.
But I don't have the app either.
That's
so I
okay.
tried to shout. Okay. What you call ionocyte subtypes, could they be ionocytes at different differentiation stages?
Yeah, that's a good question. So,
Please repeat the question.
okay, so she's asking about whether the ionocyte subtypes identified could be real subtypes or could be different differentiation stages. So as we can see in my data set, so those are from the matured cultures, and all the ionocytes we captured, we think they're kind of matured. And you can see from the heat map I showed, the gene expression signature is quite different. But I mean, could it be the other possibility could be there are different differentiation stages, but we're trying to do some like pseudo trajectory analysis on the subtypes and also on all the real subtypes as well. So that's the follow-up story on this.
I have one question, if that's okay. That is, you looked at some of the gene expression characteristics of different types of monocytes, and I noticed a number of ion transporters, but I didn't see anything for, say, the SLC26A4 or the SLC26 family that are often associated with bicarbonate transport in different models of anion transport. Did you look for those and they were absent, or did you not look for those? Or were they just in low abundance?
Okay, so the question is about some of the solute carriers or about other bicarbonate and chloride transporters could be exist in those subtypes. Actually, we didn't look into those. So we're trying to generate like ion transporter like bubble plots on all the subtypes and all different cell types. So we did check those genes, but I think to my understanding or to my data, those genes are a lot like distinct in each subtype. Yep. Yeah, if they are distinct, I would show it on a slide. Yep.
And we do have a question from the mobile app. So the question is, do you have any comment about the turnover time of ionocytes? Is turnover rapid, or once they differentiate, is the phenotype sustained?
Yeah, actually we did some turnover studies, but I cannot quite get a conclusion here. Some of the data shows the ionocytes are turnover. I wasn't sure the half time, but it's pretty like rapidly. If you take the lineage tracing like tamoxifen off, then you can clearly see those traced cell numbers drops. So yeah, it's kind of hard to get a conclusion on what is the turnover timing will be like. Yeah.
Okay, great. Thank you so much.
All right.
All right. And our next speaker will welcome Elizabeth Kramer to the stage from Cincinnati Children's.
Hi, everyone. I'm Beth Kramer from Cincinnati Children's. Thank you to the chairs and to the foundation for the opportunity to speak today. Before I forget, I'd be remiss if I didn't acknowledge my co-authors, Chris Hudak, Cindy Davidson, and JP Clancy for their help with this work. I'm excited to talk to you about smooth muscle-specific CFTR loss and how it induces airway hyperreactivity in response to TGF-beta. And I have nothing to disclose. So what do we know about airway smooth muscle in cystic fibrosis? So airway hyperreactivity, or excessive bronchoconstriction in response to stimuli such as methacholine or other noxious stimuli in the environment, are found in over half of people with CF, and we've known this for decades. And furthermore, people with AHR demonstrate a faster decline in their lung function and a greater rate of exacerbations. But of course, this is occurring in the setting of inflammation and infection, so it really was not clear to what extent the smooth muscle itself was contributing to this phenotype. We also know that children with cystic fibrosis have increased ASM burden on endobronchial biopsy, similar to children with advanced asthma. However, this was also seen in non-CF bronchiectasis. So again, the idea of maybe just inflammation and irritation can produce this, and it's not something inherent to cystic fibrosis. Then we look at studies, and there are some contradicting reports, but on this 2016 meta-analysis of some studies, it was found that CFTR mutation carriers, who we don't tend to think of as having excessive inflammation or infection, have an increased risk of asthma. And finally, Dr. Stoltz's lab from Iowa has shown that newborn CF pigs, prior to the onset of infection and inflammation, have airway obstruction and airway smooth muscle constriction. And they hypothesized that CFTR, which they found localizing to the smooth muscle in the sarcoplasmic reticulum, contributes to proper ion charge balance. And without it, calcium is not properly uptaken into the SR. So all of this put together makes it seem like there really is a smooth muscle problem in cystic fibrosis that goes beyond just what we would expect due to chronic infection and inflammation. But what causes this? And whenever I think about a lung disease phenotype or a symptom phenotype, I think about this graph, and I love it. It's, the inputs into the final phenotype, right? You have the CFTR genotype, you have random events, environmental factors, and of course, genetic modifiers of cystic fibrosis. But we probably need to think about that just beyond the epithelium and more in depth. So for an example, this is a slide, a piece of histology from one of our mouse lungs. It's stained with alpha SMA in brown. So here we have an airway with smooth muscle around it, a vessel. There's some inflammation. You can see the alveoli out here. And when we think about the inputs into the phenotype, the problems with these different tissues, we should think about it in terms of the inputs into the epithelium, the inputs into the smooth muscle, and probably across different organ systems and different tissue types that I'm not even going to go into today. And why does this matter? And I think it bears mentioning for a moment. Why do we go beyond the epithelium? Well, I think it does matter. So when we think about assessing clinical benefits from therapies, how we design our trials, what are our endpoints? Should we just be looking at sweat chloride, or should we go a little bit more in depth? When we think about residual symptoms, that may become apparent in people with CF in this changing landscape as they live longer lives. And it's also important when we think about gene therapy targets. If there are fundamental defects in tissues that we're not correcting with gene therapy, then we should expect those symptoms to persist. So this matters. So let's go back to the genetic modifier idea. So TGF-beta, transforming growth factor beta-1, is a well-known genetic modifier of CF. But I'm going to take it a step back. So what is TGF-beta? It's perhaps best known as a pro-fibrotic cytokine, but at lower doses, it's important in inflammation, it's important in injury, response, and repair. We know that naturally occurring higher producing TGF-beta polymorphisms are associated with a greater risk of asthma and a greater risk of developing severe asthma. We also know that if you take human precision cut lung slices and drop TGF-beta on them, they contract. So I think all of this comes together to say there's a pretty good evidence that TGF-beta is important in airway constriction. How about in cystic fibrosis? So this is a study from Dr. Drum looking at, it's a genome-wide association study looking at genetic modifiers of cystic fibrosis. And those same naturally occurring TGF-beta polymorphisms that cause high production that I mentioned are risk factors for asthma were also found to be risk factors for developing more severe CF lung disease. So here, these are two high-producing TGF-beta polymorphisms, and in red are individuals who had the more severe phenotype, and they are overrepresented among people with those high-producing polymorphisms. So some pretty good data that TGF-beta is important in cystic fibrosis. And Dr. Harris from UAB has shown that TGF-beta in both the blood and the bronchoalveolar lavage fluid is a biomarker of more severe lung disease. But TGF-beta's mechanism of action in worsening CF lung disease is somewhat unclear and probably multifactorial. We hypothesize in our lab that TGF-beta drives CF-relevant pathology and also that CFTR deficiency enhances susceptibility to the detrimental effects of TGF-beta. So when studying TGF-beta, the first difficulty was how to make a physiologically relevant model. There are transgenic mouse models of TGF-beta overexpression available. They develop rapid obliterative fibrotic lung disease, which was not what I wanted to study. So we turned to an adenoviral vector that expresses a constitutively active TGF-beta transgene, and that lets us titrate TGF-beta expression in the lungs. In this study, I'm going to show you some background data before I get into our new data for today. This is in the Delta F508 homozygous mouse model. And I put an asterisk here to remind you that the CF mouse does not recapitulate human CF lung disease, right? They don't develop that mucus oversecretion phenotype. But with proper stimuli such as Pseudomonas infection, age, cigarette smoke, they do develop relevant pathology, and we have powerful genetic tools in this model. And as control, we have heterozygous carrier mice as non-CF mice. So these mice are administered the adenoviral vector containing TGF-beta, or as a control, PBS, or an empty vector control. And I will note that at its dosage, the empty vector does not produce inflammation or other significant effects by itself. And this was to kind of get us in this physiologically relevant range. And so first what we noticed is in what I'll call the CF mice here, we saw excessive goblet cell hyperplasia. So they developed mucus plugging. So this is a slide showing a branching airway, and here we've caught some mucus there. So they just developed excessive goblet cell hyperplasia after exposure to TGF-beta. We also looked at smooth muscle. So again, this is alpha SMA staining in brown, and we quantify the area of smooth muscle around the airways. And you can see that the CF mice develop significantly more smooth muscle in response to TGF-beta than do the non-CF mice. And then we took those mice and we wanted to measure lung mechanics. So we put them on the FlexEvent machine. We get some data for lung mechanics there, and I will kind of orient you to this graph here. So on the y-axis is change in pulmonary resistance. You can think of that in this as essentially a proxy for obstruction. So a greater deflection upwards when we treat the mice with methacholine would indicate airway hyperreactivity when it is excessive. So in black, we have non-CF mice treated with TGF-beta, and in red, we have the CF mice, and we have increasing doses of methacholine and then albuterol, and we got multiple measurements. I really wanted to show you the time course. These are taken about every 12 seconds on the cycles, the time course of the lung resistance here. So you can see, here's our low dose of methacholine. They reach a pretty similar point midway through, then the CF mice kind of keep creeping up. When they have the high dose methacholine, the CF mice kind of plateau at this high level, and then kind of have a semi-beneficial response to albuterol. But we do see airway hyperreactivity in this mouse model in response to TGF-beta. So just to summarize what I've showed you in this first part, if we imagine one of these mice airways, we have loss of CFTR function in all tissues in the lungs here, the epithelium and the smooth muscle. When we expose those mice to TGF-beta, we have goblet cell hyperplasia, we have increased airway smooth muscle mass around airways, and enhanced airway hyperreactivity. But really, that still leaves the question of what does CFTR loss specifically in the smooth muscle contribute to this? We hypothesize there's an inherent defect in the ASM that drives some of this dysfunction that we see. And really, to get at that, we turned again to a mouse model, again from the Case Western Reserve core, and we turned to a conditional null model. So this is a floxed CFTR in mouse model. This is our control mice. They express CFTR normally. However, when bred to a mouse line expressing CRE, they can specifically lose CFTR function in the tissue of your choice. And here we have chosen to drive CRE with a smooth muscle-specific promoter. So these mice lack CFTR function in all smooth muscle, but they have it in the epithelium and other cell types. And then we again administered either the empty vector control or the TGF-beta vector. So first, I just want to show you some data about how much TGF-beta we're provoking, and this is similar to prior experiments that have been published. So in both the control mice and the smooth muscle knockout mice, when we look at active TGF-beta, we see around 2 nanograms per ml in the bronchoalveolar lavage fluid. When we look at total TGF-beta, we do know that they secrete a lot of latent TGF-beta that is inactive, and we get around 30 to 50 nanograms per ml. And you can see that this is similar in the control and the smooth muscle knockout groups. We then looked at goblet cell hyperplasia. This is with the periodic acid shift stain that stains goblet cells kind of a brilliant magenta color. I've kind of shown them with the arrows as they don't always project well. And then we're able to go through and count those cells and count the non-goblet cells and show the percent of airway epithelial cells that are goblet cells. And we did not see an increase in the smooth and muscle knockout mice. And this contrasts with the CF mouse, the whole lung CF knockout mouse that I mentioned earlier. As expected, they do develop inflammation. So this is looking at total cell counts from the bronchoalveolar lavage fluid. They were trended upwards in both groups. And when I looked at differential cell counts, as we've seen in prior studies, we did see that TGF-beta induces increased numbers of neutrophils as well as lymphocytes in both the control and the smooth muscle knockout groups. But again, very similar between the groups, similar inflammatory profiles. So let's look at the airway smooth muscle histology. Again, we have an alpha SMA stain here, staining the smooth muscle in brown. And this time we really did not see an increase in smooth muscle mass in the smooth muscle knockout or in the control mouse. So no difference in our ASM histology. And then we put the mice again on the flex event machine. First, I'm just going to show you some baseline lung mechanics. So this is baseline resistance. They have not been treated with methacholine. No change in baseline lung resistance. So I've shown you a whole lot of no difference between our control mice and our smooth muscle knockout mice. Here's where it gets interesting. So this is when on the ventilator, we go ahead and we expose them to increasing doses of methacholine. And as a reminder, deflection upwards excessively would indicate airway hyperreactivity. Here I'm showing the baseline after low dose, medium dose, and high dose methacholine, and these are TGF-beta exposed control mice. And when we look at our smooth muscle knockout mice, you can see that we do see airway hyperreactivity, especially after the medium and high dose of methacholine. So these mice, despite having similar amounts of smooth muscle around the airways, still develop this hypercontractility phenotype. So to summarize this, with the delta F508 mouse model, we saw that giving increased pulmonary TGF-beta drives CF-relevant pulmonary pathology with effects dependent on CFTR function, and specifically in that whole lung loss of CFTR model, we saw increased goblet cell hyperplasia, increased airway smooth muscle mass, and airway hyperreactivity in the CF mice. To contrast that, when we have loss of CFTR function only in the airway smooth muscle cells, we see a slightly different phenotype. We see not excessive goblet cell hyperplasia, not excessive airway smooth muscle mass, but we still have that residual airway hyperreactivity, indicating that likely enhanced airway hyperreactivity in response to TGF-beta is due to defects inherent to CF airway smooth muscle. The contributions of the airway epithelium and immune cells and other cell types are unclear at this point. Likely, I would hypothesize that the epithelial CFTR loss contributes to goblet cell hyperplasia that we saw in the delta F508 mouse model that we did not see in this smooth muscle specific mouse model. In the future, I'd be interested in looking at epithelial-specific knockouts to see the contribution of the epithelium, to study in vitro co-culture systems to look at the mediators and the conversation between the different cell types, and finally to investigate other triggers of airway smooth muscle dysfunction in CF. I'm under no illusion that TGF-beta is the only thing that does that, is simply the hammer that I'm using for this model, and I'd like to see what else can do this as well. So thank you everyone for your attention and for the supporters, and I would be happy to take questions over the app or just shout it at the podium. Yeah. Right. I have not. I think that's a really interesting idea, but I have not looked at that. It does, TGF-beta is one of those molecules where it does almost everything, but I think it's a good point. Thank you.
In at least some of the other animal models of CFTR knockout, there's a reduction in smooth muscle associated with hollow organs. Do you think the change in smooth muscle, just overall mass, is tissue dependent, CFTR dependent, TGF-beta dependent? How do all those things come together? And by the way, in some of those other ones, TGF-beta seems to exacerbate it and make it fall apart faster.
I would bet it is organ dependent. We certainly see variable effects in different organs in the knockout mouse. And thinking about smooth muscle, I mean, certainly, for example, the tracheal smooth muscle contraction is disorganized and less so than in wild type. So they don't have a hypercontractile phenotype. They have kind of a very disorganized contraction that is weak. So I do think it probably is tissue specific. I think there's probably tissue specific mediators. I'm very interested in as I'm learning more about abnormal Th2 type of inflammation and cystic fibrosis, I think there probably is something going on in the lung, and the crosstalk between the epithelium and the smooth muscle, that's pretty relevant here.
We have another question on the mobile app. So do you have any comment about the That was the last one. Did you see any small airway remodeling or change in the distal airway morphometry, such as change in airway number or size in the stimulated CF mouse?
That's a good question. No, we did not. And we did go through and we quantified the sizes of airways and also in more intricate studies compared that to the smooth muscle around the airways, since certainly in the normal mouse you have different amounts of smooth muscle mass depending on the caliber of the airways. And we did not see any changes in either of the CF models that I've discussed today.
I do have one quick question, just out of curiosity. Do you think any of the currently available modulators address some of this?
That's a great point. I think the answer is likely yes. So there was a study, I believe, from the Stoltz group looking at folks with G551D treated with ivacaftor for only 48 hours, and they saw a reduction in air trapping and rapid improvement in FEV1. And I would bet some of that initial improvement in FEV1, the very rapid improvement, is not just that purging of the mucus, but also some improvement of smooth muscle caliber. And I believe there were some studies looking at smooth muscle around vessels, and that there seemed to be a relaxing potentially of the vascular tone as well. So I do think that likely modulators help. How much, I don't know. You know, it would be interesting to take a lot of kids before and after modulators and give them methacholine challenges, but it's not something we clinically do especially often. But if anyone wants to try, you know, send me the data after you get it.
Thank you.
Thank you.
Great. And then next, we'll welcome to the stage Trevor Perry.
Updated slides came through, okay?
So am I. You're set.
Cool. All right, thank you so much for the opportunity to speak here. My name is Trevor Perry. I'm the VP of Research at Crystal Biotech, Inc. We're taking a little bit of a left turn in terms of the topic of today. And so I'm actually going to be talking about use of primates to understand at least some aspects of clinical delivery of our gene therapy vector KB407. So for the disclosures, I do have disclosures for this talk. So all of the authors on this abstract are all current employees of Crystal Biotech, and we all have an equity interest in the company, and the company did pay for all of the research that's shown here. I will not spend any time educating you on cystic fibrosis as a group. The one thing that I wanted to emphasize, and obviously something I think that was really important in the plenary, is that we're really interested in finding therapies for those who are not amenable to modulator therapy, whether that be because of their genetics or because they can't tolerate it. But there is certainly a subset of CF patients that don't have any treatment available to them to date, and we're interested in developing one. And we're doing it in the context of gene therapy. So a little bit of background on what our vector is. So KB407 is a gene therapy vector based on herpes simplex virus type 1, the type of virus that causes cold sores. We have engineered our vector to be replication defective and reduces cytotoxicity as compared to the wild type virus. And there are a couple of kind of interesting benefits about HSV1 as a vector that we think may be helpful in developing a CF gene therapy. So one of those is that it has a very large genome, in relation to other viruses. So the genome for HSV is greater than 150 kb, which allows for a very large packaging capacity of foreign genetic material. And then so CFTR is a relatively large transgene, about the limit that you could put into an AAV vector, whereas for KB407, we actually have 2 full-length copies of the gene incorporated into our vector, so we make full-length CFTR protein from it. In one of the other really interesting aspects about HSV1, it's prevalent in the human population. About 2/3 of the human population actually have HSV1 infections. And interestingly, there's a very sophisticated mechanism of immune evasion by HSV1. And that's why I say, after probably 40 years of effort, we still don't have a good HSV1 vaccine, because the virus is really evolved or co-evolved with humans for hundreds of thousands of years and really effectively evades the immune system, which then allows us to redose our vector. So we actually don't have some of the same issues with immunogenicity upon either baseline immunogenicity or development of immunogenicity over time. And this has really been borne out in a validated phase 3 study that we've completed in our lead product that is a topical gene therapy for a different disease called dystrophic epidermolysis bullosa. But in that context, we actually dosed these subjects weekly for 6 months with our vector. And we dosed in subjects that were anti-HSV1 positive at baseline and anti-HSV1 negative at baseline. And we see great efficacy upon redosing over time. And we don't see any impact on anti-HSV antibodies on efficacy of our drug. So we don't think immunogenicity is a significant issue for HSV1, which is great. One of the other interesting aspects about HSV1 is it lives episomally. So when it enters the nucleus of the cell, it does not integrate or otherwise interact with the human genome and is completely separate from it. So there's really no risk of kind of oncogenesis that people are concerned about for insertional mutagenesis. So we've used this vector technology that we've developed, and we've inserted the CFTR transgene and developed our product KB407 for targeted gene delivery into the airways of full-length human CFTR. And we have formulated it in such a way that you can actually nebulize the intended phase 1 clinical dose in less than 30 minutes, since nebulization time is an important aspect to keep in mind for treating a CF subject. So I'm going to give you just like 3 slides of background information that we presented previously before I really delve into the primate work. One of the slides, so here we showed that the CFTR that's encoded in our vector is actually functional. So we used the 3D patient-derived organoid model to establish this, looking at correction of the CF defect in organoid swelling in both class 1 and class 2 organoids derived from patients harboring class 1 and class 2 mutations. So we do know that our vector delivers functional CFTR that can fully correct the organoid phenotype within 24 hours of transduction of those cells. We had conducted a single-dose safety study of our vector in mice, both healthy mice and the gut-corrected CFTR knockout mouse, and we showed that the vector can be delivered to the airways of mice with the nebulizer safely. We also get good delivery of our vector, both the genome itself as well as the transgene, into the airways of mice. We then moved into what's shown here, which is a feasibility test of redosing of our vector in non-human primates. So this is 2 doses of KB407 delivered by nebulization using what we intend to use in our clinical trials. The dosing was spaced 14 days apart, and we looked at 2 days after last dose administered, we looked at all aspects of safety of delivering an HSV1 to the airways, as well as looking at where does the virus go and can it express the gene of interest in the cell types of interest. And so what we found was that when you nebulize KB407 into a primate, it distributes equally across the airway and doesn't really go anywhere else systemically. So it's really a localized delivery of the CFTR transgene into the airways of the primate. And on the right is actually QRT-PCR data looking at expression of our human CFTR delivered in the lungs of monkeys. So this kicked off a large non-human primate toxicology study where we were repeat weekly dosing primates using the clinical nebulizer, but to establish the toxicology profile of our product before we moved into the clinical testing. So here we delivered either low or high dose KB407 or vehicle control or air control weekly to these primates. And then we took down a group of animals 1 day after last dose administered, and we took down another group of animals 28 days after last dose administered. We looked at all assessments of safety, food consumption, body weights, clinical observations, pulmonary function, cardiac function, eye physiology, macroscopic observation at necropsy, microscopic observation. We really see no significant safety findings on repeat dosing of our virus. And actually, what was really nice to see on the right here, this is in situ hybridization, looking at the human CFTR transcript expressed from KB407 in the primate lungs. And we can see, looking here in this set of images, this is actually 28 days after the last dose of 407 is administered, and we can actually still see expression of the CFTR transgene in the primate lung upon nebulization, 4 weeks after dosing. So, we know that we're getting into the primate lungs, and we know that we're expressing the CFTR, but the question really is, what cell types in the primate lungs are we getting to? And are we getting to relevant cells that might be suggestive of actually having some sort of physiologic, disease correction of the defect? So that brings to really the kind of meat of this talk. So what we did was we took sections from the high-dose toxicology animals who'd received 3 weekly doses of KB407, and we did cell type specific staining for relevant CFTR, endogenous CFTR producing cells, so in this slide, we're looking at AC tubulin staining, looking at the ciliated cell population, and we wanted to say, is our virus getting into these cells? Unfortunately, so these are healthy monkeys that have their own primate CFTR, and primate CFTR and human CFTR are very similar. They're 98% identical at the amino acid level. So we can't actually stain specifically for human CFTR in the primates. And so what we do is we actually use a surrogate marker for expression from our vector. So when our vector transduces primate cells or human cells, there are actually only 3 HSV genes that are expressed from our virus. All 3 of those actually contribute to immune evasiveness. And we've actually selected one of those proteins for immunofluorescence to identify which cells are actually transduced successfully by our vector, trafficked to the nucleus, and actually expressing genes therein, which is shown in red here. So we actually took from sections across these high-dose animals as well as the vehicle and air control, we took 10 fields of view, non-overlapping fields of view with this co-staining process, and then manually counted the total number of ciliated cells in this set of imaging, viral marker positive cells in this imaging, and then looked at what are the percent positive ciliated cells upon KB407 therapy. And so what we can see from our NHP study is about 60% of all of the ciliated cells quantitated in this study were actually KB407 positive. So we actually transduced a majority of the ciliated cells in the conducting airways of primates. We then did a very similar assessment to this, but here we're looking at club cells, or standing for SCG B1A1. Again, same viral marker, looking in the controls versus the high-dose animals. And we performed a quantitation, and we see about 17%, 17.5% of all of the club cells are actually also transduced by KB407. Similar story here. We're looking at MUC5AC, looking at goblet cells in these primates. We see fewer total goblet cells in the lung sections. And so we see about 8% of the goblet cells are also positively transduced with KB407 upon nebulization. And one question that we get asked a lot is, well, what about macrophages? Is most of the virus that you're nebulizing into these monkeys actually getting taken up by resident immune cells and destroying your vector and not actually delivering the gene of interest to cells where it can actually be successfully expressed? So we did another staining experiment where we looked at co-staining with CD163, so a marker of resident lung macrophages, and we looked at the total number of 407 positive cells that were also positive for CD163, and we see only about 20% of the cells that are 407 positive are in macrophages. So we think obviously a large majority of the cells that are transduced by KB407 upon nebulization are not being taken up by immune cells, which was very pleasing to see. And we also looked at basal cell transduction with our vector. The basal cells, for the most part, are not accessible to the air, and so we did not really expect to see transduction of the basal cells, although obviously it would be great if we could, but because we're an aerosolized delivery, we didn't suspect to see basal cell transduction, and we certainly did not see colocalization of the basal cell marker in 407. So 407 is really limited to transducing the air-exposed cells in the conducting airways. It actually, one of the great things about HSV1, it has a very broad tropism to many human cell types, and we actually see the tropism in the airways is also pretty broad, where we can see transduction of secretory cells as well as ciliated cells. And I think another relevant question for this was, well, how much CFTR are we expressing in these primate lungs if we can actually get into them? So here we took lung biopsies, and we did an absolute quantitation of the total human CFTR transcripts versus the total endogenous primate CFTR transcripts to get a kind of a sense of, are we in a therapeutic range of correcting back to a reasonable endogenous level of CFTR, at least mRNA, in this nebulized primate model. And what we can see actually across lung biopsies is that we see somewhere between 100 and 400% of human CFTR expression versus primate CFTR expression. So in the cell types that we hit, we actually can express at or above endogenous levels of CFTR in the primate lung using the clinical nebulizer. And I think one other question that we get asked a lot, so moving to another type of primate, which is human, we are looking here at inflammation induced by our virus. So one of the things we think that's great about our vector is it really doesn't mount significant inflammation that's been seen throughout our clinical development of other programs. as well as doesn't mount significant immunogenicity. And so we were interested to see, does transduction of CF primary human epithelial cells induce cytokine expression at a significant level? Are we seeing acute inflammation from these cells after we transduce them with KB407? So to discuss what this data is, so on the bottom left, there are actually 4 genotypes of cells. And so all this data is actually the average result in those 4 cell types that are transduced either with a vehicle control or KB407 and multiplicity of infection of 0.4, 2, or 10. And so the top left here, we're looking at QRT-PCR, so expression of the human CFTR mRNA coming from our virus in the human cell types in culture. We can see definitely these human cells are getting transduced by our virus and are expressing the CFTR. So we know the virus is getting into the cells. We then did a multiplex ELISA assay to look at a whole bunch of inflammatory markers, IL-1 beta, we look at TNF-alpha, interferons, alpha, beta, gamma, and what we can see is there is no significant induction of inflammation from the CF primary airway cells as compared to the vehicle control. And here we're using poly-IC, which is a synthetic double-stranded RNA, as a positive control to induce inflammation. So we know that these cells are reactionary to kind of like an antiviral type immunity in terms of cytokine expression, and we just don't see induction of any of the inflammation from these cells after transduction. So to provide a brief summary of the data, so the top data points that are kind of shown in gray are things that we've shown previously. Actually, a lot of this was from last year's NACFC. But in blue here, so we've shown that our vector, using the clinical nebulizer in primates, we can transduce and express CFTR, or we can transduce ciliated cells, goblet cells, club cells, so endogenous CFTR producing cells in the primate airways. Those cells can express genes from the HSV1 genome, so we know we're seeing successful delivery into the nucleus and expression from our gene therapy vector. We're correcting to at least endogenous levels of CFTR transcripts as compared to primates in this testing, and we are not inducing significant cytokine or chemokine expression in transduced primary CF patient airway cells. And so, we've been working diligently with the FDA. We actually filed an IND application, which was accepted in July of this year, and we're working on kind of site startup activities, working towards a phase 1 trial of our gene therapy vector. So with that, thank you very much for your attention. Thank you for the Crystal folks who all contributed to this, and I'm happy to take any questions. Mm-hmm. Yep. Yeah, so, sorry, the question is, do we have functional data in these human cells for CFTR? Yeah, so we do have the functional data from the 3-dimensional human patient CF cells. From these cells, these are actually submerged cultures, not ALI cultures. So we haven't done the oosing study with these. Sorry? Yeah, so we're working diligently on doing the ALI oosing type studies. I think one thing that we talk a lot about in thinking about oosing studies is that the ALI model that was really developed to show small molecule correction of a defective CFTR in those cells. It's been successful, obviously, with the modulators, and so we're trying to understand when we're delivering just a complete separate full-length copy of CFTR in the mRNA. Is that the appropriate model to show function? We're trying to understand that. It's been difficult to transduce the ALI cultures, has been the challenge that we're working with, and I think we're not alone in the gene therapy field finding difficulty transducing those cells, but it's certainly something that we're working towards, particularly because we know we're getting transduction in the submerged cultures from patient cells. Right. Yeah, and it's something that we certainly, we get asked about, and we were really working diligently towards, and can we modify this experimental system, take these to ALI, and then do an oosing study after kind of full differentiation. We're working with a collaborator in Newcastle, Dr. Keiko, on those exact questions right now. Right.
I
Yes.
do,
Oh.
sorry, I just want to read, because there was one that came in early, from the app. What happens, or sorry, they keep coming in. What's the percent transduction with HSV vector and epithelial cells other than ciliated ones?
So
The earlier question.
the only quantitative data that we've looked at in the airways are ciliated club and goblet cells. And so we haven't looked at, in the same kind of quantitative context, any other cell types in the airways other than those.
Since you have no immunology for the HSV itself, does the human CFTR have a limited signal in the lung?
So we did not assess anti-CFTR antibodies in the primate. I think it's difficult because primate and human CFTR are so similar. You might not expect a response because they're 98% identical, but you are delivering a foreign protein into an animal model that has fully immunocompetent. So is that, if you did see a response, a CFTR antibody response in primates, is that reflective of what you might see in a human subject? We don't know. Right. So yeah, and we did do the primates because we think that's the closest thing we're ever going to get to a human in the non-human, in non-human testing, and that's why we pursued it. But we have not looked at CFTR specific antibodies. There's actually not commercially available kits to actually do that. We are working on developing an assay specifically to look at those that question, and we have samples to test, but it's not simple to do. So it's something we're actively pursuing.
Have you tested to see if your non-human primate is making small amounts of vector protein itself? But you even test to see that it's not making any small amounts of the viral protein?
So we do know that when our virus transduces primate cells or human cells, there are 3 proteins that are expressed, definitely, upon infection. And it's part of the expression cascade of HSV1. There are 3 groups of genes: immediate, early, and late. And so we do express 3 of the 5 immediate early genes, but we've knocked out 2 of them that then does not allow for expression of the earlier late genes. So those 3 are expressed, and we use one of them as a marker in the primate lungs. We've done QRT-PCR looking at expression of early and late HSV genes to see if the mRNAs are even present in animal models when we dose. We don't see expression of those kind of later genes, which we expect given the engineering that we've done. So to the best of our knowledge, there are only 3 HSV proteins expressed in human subjects upon transduction with our vector products. Yeah, so we as part of the, in the in situ hybridization data that we looked at, we've looked at both day 1 and day 28 after dose, and you definitely see a diminishment of the signal at day 28. So we do decrease signal over time, and we think likely that's driven at least in part by cell turnover. We do see a slight reduction actually, so we've done qPCR for genome copies in the primates at both of those time points as well. And we do see a slight reduction of the genome copies in the primate tissue. So we do expect that the vector is getting turned over likely through the process of cell turnover in the lung epithelia. So there is kind of a time-dependent loss in signal detection. And so that's why we think, obviously, redosing is going to be a critical aspect of our treatment kind of moving forward. So we haven't talked about what the promoter is for our vector. So we've done lots of promoter, so not specifically in the lung context, but we've done lots of promoter work looking at endogenous versus exogenous and synthetic promoters and kind of persistence of gene expression. So that is work that we've conducted as part of vector development as a whole for our company. Yeah, we think it's likely attributed to turnover of the vector itself, probably because you're seeing cell turnover over time.
We are unfortunately going to have to move on, but one last quick question that did come through was, how does HSV cross through mucus plugging?
Yeah, so a great question. We have done an in vitro mucus assay. We have not tested our vector in these kind of CF animal models, which, as you know, are hard to come by. And actually, the cross-species transduction efficiency of HSV1, it doesn't work in pigs, so we can't do it in the pig model. And I think the ferret model is an open question. But we've looked at it in vitro, where we looked at the mucus penetration efficiency of our vector through primary CF patient cells that were allowed to accumulate mucus on their apical surface for 7 days, and then we put the virus on top of the mucus. Or we washed the apical surface free of mucus within an hour of transduction, and there actually is no quantitative difference in transduction of the underlying epithelial cells with an hour of mucus accumulation versus 7 days. So we don't think it is a absolute barrier to transduction with our virus. Obviously, it's an open question in the field. It's something that we are very interested in seeing in a phase 1 study.
Great.
Thank you very much.
Thank you. Great. So now we'll move on to the next talk, and we'll welcome Lawrence Rasmussen to the stage. He's coming from University of Alabama at Birmingham.
Thank you for the kind introduction, and thank you all for joining me here. I'm excited to share my research. There we go. Title, PDE4 inhibitor of Primulase enhances benefits of CFTR modulators both in vitro and in vivo. So I do have one disclosure, Celgene, which helped spark back in 2017 to 2018 the inception and idea of looking at a Primulase and its possibilities as a CF therapeutic. So before I dive into the data, I do want to talk a little bit about this current state of CF therapeutics as they are right now. Definitely revolutionary were the CFTR modulators that target the defective CFTR protein rather than the disease manifestations. However, while many patients with CF benefit from triple modulator combination therapies, unfortunately, maximal clinical benefits are not achieved in everyone. So that's going to be one aspect I will be examining, is how to increase the actual benefits of CFTR modulators. And another area I'll be looking at for the CF is addressing chronic inflammation, because that remains an unmet need. So when I'm talking about a potential add-on therapeutic to be added with modulators, I'm going to be considering the augmentation of CFTR modulators on CFTR, as well as attenuating the CF inflammation. So this cartoon highlights the central role of CFTR in regulating the mucociliary clearance mechanism in the airways. Let me just get the laser pointer here. Perfect. So I want to highlight this particular enzyme, phosphodiesterase. And I do apologize, there were some issues early on, and I give a lot of credit to the IT people of this symposium for helping me get this to where it is now, but some of the wordings and fonts will be a little bit off. So right before that, number 4 should be phosphodiesterase, and phosphodiesterase should be PDE. So PDE is an enzyme that controls and regulates cyclic AMP, and if we were to inhibit it, which I'll be talking about in a few slides, will actually increase the cellular cyclic AMP, which will drive the primary route of CFTR activation through protein kinase A. So just picture the previous slide with this title. And I know this slide is a little bit busy, but I think it highlights very well that PDE4 has been well studied in the context of inflammation and has been shown to influence several signal transduction pathways that modulate inflammatory responses. And on that, PDE4's connection to attenuating inflammation has resulted in many PDE4 inhibitors to be evaluated for several inflammatory diseases. And focusing just on the lungs here, many of you may be aware of roflumilast, which has been prescribed to help treat people with chronic obstructive pulmonary disease with symptoms that are very similar to patients with CF. So given the potential of PDE4 as a therapeutic target, we wanted to evaluate what it could do in cystic fibrosis. And there's been some data previously published by very respectable groups. Dr. Hannaran right here has shown that in delta F508 primary CF human bronchial epithelial cells, HBE cells, when looking at a wide array of phosphodiesterases in airway epithelium cells, in the CF cells, PDE4 is is expressing a higher gene than the others. Dr. Blancard's group has shown that PD4 is actually co-localized with CFTR in airway epithelium, as we can see in both their immune assay, where we see CFTR and PD4, and a pull-down assay where roloprem, which is a first-generation PD4 inhibitor, helps prove that PD4 is near CFTR. So given all this information, our group wanted to evaluate PD4's apremilast capacity to help patients with CF. One thing I would like to highlight is apremilast is already FDA approved to treat inflammatory skin diseases such as psoriatic arthritis. So when I'm evaluating apremilast, I'm going to be looking at three particular areas: airway CFTR activity, mucus physiology, and lung inflammation. So to begin, I tested whether, one, can apremilast actually activate CFTR? And two, what is the capacity of apremilast's ability to work with CFTR modulators? Is there any sort of cross-chemical interactions that would negate the benefits of one or the other or both? So in wild-type HBE cells mounted in Using chambers, I found that even following ivacaftor apremilast could still activate CFTR further, as we can see in both the Using chamber tracing on the left and the bar graphs on the right. So building upon these results, I tested what additional benefits apremilast could provide in primary CF-HBE cells that were previously treated with modulators, as tezacaftor, TEZ, elexacaftor, ELX, or tezacaftor plus elexacaftor. And as seen here again in both the Using chamber tracing on the left and the bar graphs on the right, apremilast provided a significant increase in CFTR activity in all treatment groups. So this is very good. We've shown that it can work and activate CFTR and extend the benefits of modulators. But does this translate to meaningful improvements in the mucociliary clearance system? So using micro-optical coherence tomography imaging system, which is a laser that's able to capture several parameters of the mucociliary clearance system, such as the ASL depth, the PCL depth, which houses the cilia, as well as the mucus viscosity, we're able to see that immediately ASL depth is increased in the cells, in primary CF-HBE cells that were given the triple modulator therapy, ivacaftor, tezacaftor, elexacaftor, with apremilast, as denoted by this red bar. But I do want to draw your attention to this yellow square, which highlights the area of mucus viscosity. So viscous mucus will appear very bright, having a higher pixel intensity. And when we actually graph this through a histogram, as seen in this middle diagram, we see that the cells that were given apremilast actually have a reduction in the area under the curve of the histogram. And we believe this is in part the reason why we see an increase in mucus transport right here. So far, I've illustrated that apremilast can augment the benefits of CFTR modulators in CF cells, and now we wanted to look at an in vivo CF rat model. Unfortunately, there are no current CF rat models possessing a human homozygous delta F508 CFTR mutation that can respond to modulators. Thus, I used a CF rat engineered with a human G551D CFTR mutation that does respond to ivacaftor. So with these rats, I performed a nasal potential difference, which is measuring the voltage change across the nasal epithelium of the CF rats under conscious sedation. And as we can see in this electrophysiological tracing, as well as the bar graphs, when I infuse the nasal airway cells with ivacaftor, we see an increase. But we also see a significant additional increase in CFTR activity when we infuse with apremilast. And I included forskolin here to show that the ceiling hasn't been met yet in terms of the potential for CFTR activation following apremilast and ivacaftor. So again, I wanted to see, do these benefits on CFTR activity translate to improvements in mucociliary clearance system? And I wanted to look at it in the context of CF rats that were challenged with LPS, something that could induce inflammation. So here, after I've treated the CF rats for 3 days with vehicle, ivacaftor, apremilast, or ivacaftor and apremilast once a day for 3 days prior to their 24-hour LPS challenge, and taking the freshly excised trachea from these rats, and again using the micro-OCT imaging previously shown, I looked at the perciliary liquid depth, the mucus transport, and the mucus physiology changes. And we can see that rats that had received apremilast had an increase in PCL depth, an increase in mucus transport rate, as well as a reduction in mucus physiology or mucus viscosity. So in both in vitro and in vivo CF models, I've demonstrated apremilast's ability to augment modulator benefits on CFTR and mucociliary clearance. But one aspect we really wanted to delve into is that second portion of what we're looking at as an add-on therapy. Can it help attenuate chronic CF inflammation or a CF inflammation? So since this remains an unmet need, I took the VAL, the bronchial alveolar lavage fluid from the previous LPS challenge rats, and did a total cell count looking at the immune cells. And we can see that, interestingly, apremilast does attenuate both the overall immune cells as well as the percentage of neutrophils with ivacaftor. And I'm not quite sure why this mechanism exists, but if anyone has any ideas, I'd love to discuss further. And we see something very similar when we do H&E slides looking at these lungs of these rats. And as we can see here in the vehicle tray rats, we have a high amount of immune cells indicative of our inflammation model. And we see a little bit of reduction with LPS and apremilast, but again, we see a very strong reduction when both ivacaftor and apremilast are combined. So lastly, I examined 28 cytokines using a Luminex assay in our LPS challenge rats, and many of the cytokines were, as I would expect, significantly decreased as a result of apremilast. Apremilast has those anti-inflammatory properties, and this looked very good. But again, interestingly, for some cytokines, there was a significant reduction when apremilast was combined with ivacaftor. So in summary, apremilast can enhance CFTR modulator benefits on CFTR-mediated mucociliary clearance in both CF delta F508 homozygous cells and CF rats possessing human G551D CFTR mutations. In addition, apremilast demonstrated the capacity to attenuate neutrophil inflammation in LPS challenged CF rats, particularly when combined with ivacaftor. So taken together, this data indicates PD4 inhibitor apremilast may provide meaningful improvements for patients with CF currently on CFTR modulator therapies. And with that, I'd like to thank everyone who helped make this possible, Dr. Vamshi Raju's lab, the Cystic Fibrosis Research Center at UAB, Dr. Steve Rowe, Dr. Burkett, Dr. Gagar, the UAB Microbiology Department, the funding support, and again, I want to thank the IT here who helped make this possible, because when I first uploaded all my slides, it looked great on my Mac. All the color disappeared when I saw it here. So I was scrambling early this morning. So with that, thank you very much, and I'm open to any questions. Yes. Mm. So they were on treatment for 3 days. Yep.
Do you expect to see a reduction in inflammatory markers?
It's a good question. It's something I would like to see. Whether I will see it, I don't know. I need to do that.
So the various PDEs have different profiles of expression throughout the body and in tissues, and just humor me for a while and help me understand what are the major sites where PDE4 is expressed. I was thinking it was expressed in cardiac tissue too, and so I'm kind of curious about treating patients with this?
That's a very good question. So there are, just as there are many PDE4s, as there are phosphodiesterases, there are some isoforms of PDE4s as well. And the particular one in the airway epithelium is PDE4D. And that isoform is the one apremilast predominantly inhibits. It does have some off-selectivity inhibition to some of the other PDE4s, but it's predominantly targeting the D isoform. A very good question.
I also have a question. Just in the cell model, you talked about the F508del mutations, but some of the data suggests that apremilast alone can be helpful. So I was curious if you looked at any other mutations.
No, that's a very good question. One of the goals for myself is to find people with other mutations, other models, and to examine it in there. We've only looked at the Delta F508 and the G551D, but anyone who's interested, I am willing to discuss further. Thank you.
We do have a question from a couple of questions now coming in. Does apremilast decrease inflammation in an already infected animal where the baseline inflammation might be much higher?
That is a good question. I have not done the experimentation to actually answer that, but it is a good question, one I would like to examine in the future.
And is there any inflammatory marker that can be examined in human CF-ALI cultures? Would you expect similar results without the immune system?
Another very good question. I would actually have to research that more. All right, thank you.
Thank you. Great. And now for our final speaker, we'll welcome Allison Lucina from the University of Iowa.
Hello, my name is Allison Lucina, and today my topic will be the optimization of the CFTR-G551D ferret model for early synchronized lung microbiome colonization. I'm with the National Ferret Resource and Research Center, as well as the University of Iowa. As Dr. Engelhardt and many of my colleagues have discussed in the past few weeks, the ferret model presents with an unparalleled opportunity to assess CF multi-organ disease due to similar pathophysiologies in the lung, pancreas, and GI system. Additionally, ferrets are the first non-rodent large animal models with the ability to perform CRISPR gene editing. While I have had the opportunity to work with many individuals, including some of you in this room, I do not have any relationships to disclose. While the Engelhardt Lab has been creating various CF ferret models for years, today we will be focusing on the new CRISPR-mediated G551D line. This line is very similar to the model published by Dr. Sun in 2019. These ferrets mimic CF multi-organ disease, and when dosed with Kalydeco, it provides the ability to turn CF on and off. Dosage cycling and removal allows for unique ability to control CF disease progression and microbial development. Yaling Yi will be discussing more on the health and maintenance of this line in her updates in pharmacotherapy. However, today we will be focusing on lung disease progression and what we are all asking, is Pseudomonas possible in the CF ferret model? So in short, yes. Mucoid Pseudomonas was isolated for the first time in this 2.5-year-old ferret with a complex history of dosage cycling and repeated antibiotic use. The isolate was sequenced and contained characteristic MUC-A22 mutation, leading to overproduction of the polysaccharide alginate. We knew Pseudomonas was possible, but this ferret model was far from perfect. Aging CF ferrets to 2.5 years is far from realistic, and the reproducibility of this ferret's unique history was unlikely. So we moved on to the next question. Can we optimize and accelerate the model? Creating large cohorts of similarly staged lung disease would provide a pipeline for evaluating novel therapeutics. Though the primary factors leading to progression of lung disease is a common topic in not only this conference, but in CF research overall, we were posed with the question, what are the primary drivers for the CF ferret model? We studied age, sex, environmental exposure, potentiator dosage, and active bacterial transmission. With over 30 years of retrospective clinical experience, for the first time, we could do a prospective study on lung disease development in a more controlled system. However, not everything goes as controlled. So an example of this would be this CF ferret, who was born in July of 2021 and removed from VX770 at 3 months old. In December of last year, we were hit with a very big surprise, and a viral respiratory infection spread throughout our ferret colony. Wild-type ferrets were seen with minor sneezing, whereas we noted a high transmission in cystic fibrosis ferrets off VX770. Initial testing ruled out influenza and coronavirus. And thus began a very long few weeks for me and the lab, and also a less than optimal Christmas holiday. This male and many others presented with upper respiratory congestion, sneezing, nasal congestion, pyrexia, and lethargy. We learned a lot in those following weeks, including how to quickly and effectively remove nasal discharge, how to get sick ferrets to exercise. I learned a lot more on how parents of sick toddlers felt, tired and covered in mucus. And we also learned how to perform percussive therapy on ferrets. Shockingly, there is very little literature on how to perform percussive therapy on ferrets, and to my knowledge, no one is making a percussive vest that small yet. So with a little practice, we were able to find a method that proved successful on breaking up the mucus in the airways, as ferrets do not spontaneously expectorate. Symptoms typically resolved in 1 to 2 weeks, which was the case for this male as well. After the scheduled 6-month time point, we performed a bronch on this animal. On placement, we saw substantial mucus, not only in the trachea, but in the upper lung lobes as well. On culturing, this animal was positive for Pseudomonas aeruginosa. To scale up for larger cohorts, we collected an oral swab and compared it to the bowel. Unfortunately, the results were inconsistent. At his following scheduled monthly bronch, this ferret continues to have mucus, increasing CFUs, and Pseudomonas infection. I am happy to say that this ferret is healthy and that we did not lose a single animal in those very long weeks. So here is the study. Monthly bronchs were scheduled where we performed mucus assessment, CFU quantification, and MALDI-TOF characterization. We incorporated a cohort that was removed from VX770 after the respiratory pathogen to evaluate the effects of the secondary infection on disease progression. To try to accelerate disease progression, multiple methods were used for both active and passive transmission. For example, active bacterial transmission was performed by creating a composite of bowel and/or oral rinse from select CF ferrets and administering intertracheal spray. For passive methods, co-housing of up to 6 ferrets was implemented. We had high hopes that this method would increase transmission, as ferrets are social animals. They would be sharing bedding, water, food, and clearly microbiomes. Unfortunately, that cage mates did not show complementary microbial development. We also looked at potentiator cycling. However, it was confounded by the older cohort experiencing increased rates of cycling. We will continue to assess this in our future projects. And then finally, we looked at the age of potentiator removal, where we found the most promising correlation between rapid lung disease development, mucus accumulation, and Pseudomonas infection. So we dug in. We had a fairly natural break at about 100,000 CFUs per mil of BALF, so we noted that as high CFU. So as you can see in the green here, the cohort removed from potentiator at 3 months had well over a 50% frequency of high CFUs or high CFUs with mucus accumulation, whereas the inverse can be seen in the 12-month cohort. Of interest, more singly housed males had high CFUs compared to the co-housed females. However, this could be due to many variables, including weight, lung volume, and sex differences. After comparison of the microbial composition, results suggested Pseudomonas colonization occurred at approximately the same rate. There were a few cases where high CFUs were seen, but no mucus. However, mucus can be transient and abundant. First of all, apologies for the squeamish, but as we can see, we collected mucus from various animals, both pre and post-mortem. In this time, we noted a few things. Days to appearance of mucus was increased in the 3-month cohort, and we were seeing mucus in approximately half of the bronchs we were performing. On bronch placement, mucus would often obstruct the view due to accumulation in the upper trachea, as well as mucus was often thick and dehydrated, which could be very difficult to remove. Additionally, after assessing the volume of mucus, seen in the bottom middle image there, we sent lung tissue to pathology. And we found significant mucus plugging. So what you're seeing here is the airways of the animal in the right middle lobe. This animal was a 10-month-old Pseudomonas positive CF male who had been taken off potentiator at 5 months. Pseudomonas was observed as early as the first bronch. In some cases, we are unable to bronch until 6 months of age, as the ferret needs to be an adult before procedures of that nature are completed. Frequently isolated microbes included group A Streptococcus, and I know everyone has their own favorite microbes, and I am not going to list all of them here, so I'll just say stop by my poster at 6:02, and we can talk about microbes all day. Though the N number for this study is small, I believe we have the potential to create large cohorts of succinct CF-related lung disease for novel critical new therapeutics. We also have the potential to further assess longitudinal microbial development and further isolate primary factors of disease progression. Most importantly for me, though, we have a huge opportunity to add mucus to my photo albums. My desktop is currently covered. I want to acknowledge everyone in the Engelhart lab. We had a lot of work ahead of us for this study, and the animal care staff was incredibly helpful in this time. I also want to shout out Doug, because I wouldn't be here without him, and the CFF Foundation, because we all wouldn't be here without them. So with that, I'll take questions.
I was curious, the appearance of Pseudomonas, how does that impact the disease state?
Yeah, so this is it's kind of a fun topic. It very much depends on what time and how long they've had the Pseudomonas. So I have a couple animals where they've had Pseudomonas longitudinally for multiple months and don't show any symptoms at all. We will bronch and they will continue to have mucus accumulation, but no coughing, no lung sounds. However, where sometimes we'll have ones that have just gotten the infection and you can hear significant lung sounds. Usually if you get on the other end of the original infection, they seem to have a steady state where they don't produce as much concerning symptoms.
So clearly you had some cohorts here where you had them on the ivacaftor for an extended period of time and then withdrew it. Is there a clinical correlate for doing that? Is there any hope of patients being able to grow with ivacaftor and then someday not need it anymore for some reason?
Yeah, so potentially. We looked more at this like what happens if you start younger, if you start dosing younger, than what happens if you remove older. What we're finding is that if you are dosed long term in the younger years, you tend to not see that infection later in life. However, cycling was definitely something that looked like the potential to regulate health concerns. So in terms of our study, any time that we're putting an animal back on that ivacaftor VX770, we are doing it because they're experiencing additional health concerns. So anything like pancreatic insufficiency or weight loss or secondary infection, it kind of gives them the boost to be able to clear that secondary infection and then come back around on the other side. So I think that there's definitely a possibility of cycling being beneficial in the future, but it's definitely something we would have to look into a little bit more.
Remind me of the role of antibiotics here. Earlier publications, patients ferrets were on antibiotics throughout. Are these ferrets on antibiotics or just on ivacaftor and that
Yeah, so the original animal we saw with the 2.5-year-old with Pseudomonas was on cycled antibiotics on occasion. I try to avoid using antibiotics in my study unless absolutely necessary, just because we wanted to confirm that this was a possibility. So we avoided antibiotics for the most part, barring Baytril occasionally in short doses.
And related to that, you had these points about basically now the earlier removal of ivacaftor. Is there any comment here about how earlier on those animals got really sick without antibiotics? These animals, maybe on some ivacaftor, aren't able quite as systemically sick. Any comment about that?
Yeah, so what we found is, and this is kind of why we removed at that 3-month mark, that's very much adolescence for these ferrets. What we found is if we remove earlier in life, they go through a metabolic growth just at the time of weaning, which is about 8 weeks. In that time, if they are removed from the VX770 dosing, they are unlikely to thrive. So what we were trying to do was make sure that we were getting them over that adolescent hump to get to, adult prolonged lung disease. Seven days.
Please repeat the question.
When I'm talking about the short Baytril administration, typically 7 days.
Just one more question.
Yeah.
You mentioned the viral challenge 2.5-year-old. Have you also done viral challenges to younger animals to see how that may trigger or propagate?
So the 2.5-year-old was not viral challenged. It was just longitudinally in the colony. That one did experience bouts of antibiotic dosing. However, there was not a viral dosing there. In terms of trying active methods, we did not use any viral methods, but we did use innate spray back from other animals in the colony.
I had a question. I was just curious, have you looked at the airway microbiome as a whole in the ferret at all, or things such as diversity?
Yeah, so we're talking about moving forward and how to move forward in this study, and that's definitely something that we're going to be looking into moving forward. Tracheal microbiology is quite easy to get. You've got quite a bit of space there. We usually go into the left upper lobe when we're doing bronchoscopies. Just because that's a lobe we know very well, we wanted to make sure we were comfortable with the process before moving forward. So moving forward, we're hoping to look at each lung individually and how to best practice that. Additionally, we do lysates of and tissue collection and removal for microbial analysis.
Well, I guess if there are no other questions, this concludes this workshop. I would encourage the speakers to stay around, and then anyone who has additional questions for the speakers can come forward to chat with them about their work. It's been quite exciting, and thank you all for being here.